Okay, so this is Run for Your Life lesson 2. I probably should have shown you this before we started the last lesson, but this is basically the context for the unit. So if you remember, each of the units you study in biology has a context. On the wild side, the one that we previously did was all looking at global warming and how that affects organisms and so on. For this one, Run for Your Life, as you can see there, this is what comes from the syllabus. It says the topic's centred on the physiological adaptations that enable animals and humans, particularly sports people, to undertake strenuous exercise. So what we're looking at really are the physiology, biochemistry, and how that all interacts to enable people to exercise at a higher level. There's a little bit where we relate it to animals in the wild and so on, and then following on from that, we look at the medical side of it. So we're looking at a little bit of keyhole surgery and performance enhancers and so on, to see how that's all affecting performance in sport. This is what the syllabus says they want you to learn. So first of all, describing the structure of a muscle fibre and explaining its structural and physiological differences between fast and slow twitch muscle fibres. To be honest, today we're probably not going to look at this part here. We'll deal with that a little bit later on, and then we'll hopefully get on to this part, explaining the contraction of skeletal muscle in terms of sliding filament theory, which you may have heard of in sport for those of you that do sport. Include the role of actin, myosin, troponin, tropomyosin, calcium ions, ATP and ATPase, all keywords that you're going to need to be able to pick up. So what we're looking at for today, in terms of outcomes, for A grade we're looking at explain how the arrangement of microfilaments can bring about muscle contraction. For B grade we're looking at describing the ultrastructure of skeletal muscle, so really the deep structure of it, so the cells it's made up of and then what's within those cells. And C grade, so that's everybody here, recall the meaning of these terms. So fascicle, endomysium, muscle fibre, sarcolemma, sarcomere, sarcoplasmic reticulum, and myofibril. You'll see this term sarco quite a lot. Sarco tends to refer to things in muscles, so look, sarcolemma, sarcomere, sarcoplasmic reticulum, so that'll occur quite a lot. Okay, so what you need to know, there's 3 types of muscle in all mammals. You've got skeletal muscle, which is sometimes called striated or striped muscle. You've got smooth muscle, you've got cardiac muscle. Now skeletal muscle is the one that we're dealing with most. It's going to bring about your movement. So, you know, what we looked at last week, your biceps, your triceps and so on, your quadriceps, all those muscles that all the things you think of as muscles are pretty much skeletal muscles or striped muscles, striated muscles. Key point here is under voluntary control. Mostly it does what you want it to do. Smooth muscle isn't striped in appearance. It looks smooth, hence its name. You tend to find that in your organs. You've got that in your arteries. You've got that in your digestive system and so on. It's all involuntary. Okay, it's not as powerful as skeletal muscle. It never gets tired, and it's under your control. Whereas skeletal muscle, it's not under your control. Skeletal muscle is under your control, but it does get tired. Whereas smooth muscle is involuntary, not under your control, but it never gets tired. Cardiac muscles are a cross between the two, and so it's not under voluntary control, although some people reckon they can control their heartbeat, but it never gets tired, never tires out. Okay, so your 3 types of muscle. So what does it look like? So this is the structure of it, and then the ultrastructure. So each muscle, so here's your whole muscle here. Each muscle is made up of bundles of fibres. Now we call them fascicles. So you can see this one sticking out here. You can see where it says there. That's a fascicle. This bundle here, that's your fascicle. Okay. Each of the fibres then, so you can see, check out the American spelling, but each of the fibres here, and we're talking there between 0.1 0.01 millimetres across, so in diameter, but they can be quite long. So I've got here several centimetres long. Now they're formed from lots of cells that are fused together into one multinucleate cell. And what that means is multinucleate. You've got lots of nuclei all in one cell, so effectively it's like lots of cells that have joined together. Lots of nuclei in it, obviously giving it the control, and we call that a syncytium. Alright. Now the plasma membrane surrounding this syncytium, this group of cells, is called a sarcolemma. Alright, so in terms of plasma membrane, if you ever get an exam question, if it's pointing at the plasma membrane, the word they're looking for is sarcolemma. Okay, so muscle fibre here, lots of muscle fibres together make up a fascicle, lots of fascicles together ultimately make up your muscle. A couple of the things that we saw last week when we were looking at the chicken, if you remember, some of you were peeling away this epimysium here, it was like a flesh on top, but you took away the skin and there was this very thin layer on top here, and a couple of you were trying to take that off, and you could see the muscle in a lot more detail. Okay, so looking at it a little bit more deeply, so this is one fibre. Okay, so the diagram you were looking at, you had your bundles, and then within the bundles you had a bit sticking out, and then from that this would effectively have been the bit that was sticking out. So we're looking a little bit deeper now. Okay, I'm just going to rub that out, we don't need that. Okay, within one of those fibres you have these. This is, oops, need my pen working, this is a myofibril. Okay, so within one fibre you've got lots and lots of myofibrils. Notice here, nucleus, nucleus, nucleus. So it's multinucleate. Okay, this covering, this sheath around the outside, look, this is your sarcolemma, this is your plasma membrane. Okay, so really what we're talking about in this whole picture is lots of cells joined together to make one long multinucleate cell. Alright, lots of nuclei. Okay, sarcolemma around the outside. Within that you've got the bundles of the myofibrils, and then lots and lots of mitochondria. You could guess why there's lots of mitochondria. I'm sure I don't need to say that to you. We'll do a little bit more detail about that later on. Now, I put here that it appears to be banded. I said that they're sometimes called skeletal muscles striated or striped muscle, and it's because you can see, look, dark region, light region, dark, light, and so on. Okay, and there's a reason for that. If you look at each of the myofibrils, you've got different stripes here. Look, so stripes, so dark, and then in between them you've got slightly lighter regions. Okay, and that's what gives it this striped appearance. If we look a little bit closer in, now we're looking at a single myofibril. So what I just pointed out here, look, there's a dark region, dark region, dark, and then these areas here, these are lighter. Okay, and there's a reason for that. So you've got the A band, which is relatively dark, the bits I pointed at, the I band, which is relatively light. Okay. Now, if you look inside here, so see where this says actin, so it's pointing at the little blue lines, and the myosin's pointing at the little red lines. Wherever you have an overlap of both, so like this area here, this is going to look quite dark. That's why you've only got one of them, such as here on the Z disc, it's going to look light. And that overall, I suppose, gives it this kind of stripy appearance, if you get what I mean. Okay? So that's what gives the muscle its overall striated appearance. It's also really important in terms of how the whole thing works. Let me just get rid of some of this too, because these are the contractile proteins, the actin and the myosin, and the things that contract, and that's what causes the muscle to shorten, because each of these myofibrils shortens. This word here, the sarcomere, this comes up quite a bit in the exams. So let me just rub some of this out so it's less confusing. And they might ask you to measure a sarcomere, you know, sometimes asking you to do calculations and so on. They might ask you to do that. So if they do ask you to measure a sarcomere, it's the distance between two Z discs. So Z disc, the distance, that's one sarcomere. Okay. Like I said, they can ask you to calculate that, so you may have to do that. Well, measure it and then do a calculation, we'll see. Okay, so looking at a bit more detail at the actual filaments themselves, so you can see this is just expanded, so from this I band and this I band here, there's that Z disc running down, that then becomes this, and that Z disc running down becomes this area here. So in between you've got these red parts, which is myosin, and then you've got the blue parts, which is actin. Okay, so there's your myosin, there's your actin. Okay, blue actin, red myosin. Now basically what happens when a muscle contracts is that this distance here will shorten, and if you imagine on the end of each Z disc, effectively you've got more myosin here and more actin here, so the whole thing, kind of like a concertina, is going to squash in and shorten and cause the contraction. And basically these myosin heads here, look at where I'm just drawing those myosin heads attached to the actin, and like a ratchet kind of pull themselves along it, and that decreases this distance. So the distance between the two Z discs gets shorter, or what we could say is that the sarcomere gets shorter. So what does myosin look like? Well, this is one myosin head here. Okay, you've got the tail and then you've got the two heads, and it's these heads here that will attach to the actin as it contracts. They're never arranged like this picture. You always get them in this filament here, so you've got kind of bundles of them all together. So that's your myosin, that's your thick filament. That attaches to actin. This is actin, the thin filament, which is, you basically got two of these wound around each other. And you have, if you notice on the diagram, see where that's pointing at there, those little dots. They're effectively the sites for myosin to attach. And so those myosin heads that we saw will grip onto these points here, effectively, and kind of move it along. I think I said to some of you last week, it's kind of as if you imagine passing a ladder over your head if you're all stood in a line, and your arms are like the myosin grabbing the rungs of the ladder to pass it over the top of your head. Kind of how I envisage it anyway, but I'm sure your minds probably work slightly differently. Okay, so appearance of muscles. So this is just a summary. At this point now, hopefully, you can have a bash at these questions in your head. So if you want to pause the video, and you can see, can you answer these? So why do muscles look straight? Which filament is known as a thick filament? Which one's a thin one? And then briefly, in your words, try to describe how the filaments bring about the contraction of a muscle. And you want to use some of those keywords that we talked about in order to do that. So you're going to pause the video. What you can then do, if you like Prezi, I've set a Prezi up that does the next part. If you want to take that and type that into your browser, there's a Prezi which you can run through, which has got a few videos in it and a few links, so you can have a look at that, and that'll show you how muscle contraction works. But I'm just quickly gonna go through the rest of it using this video, so that you can try to get your head around it that way. Okay, hopefully you've had a go at those questions. If you've managed to include these keywords from the C grade outcomes, then you've done pretty well. Hopefully you're a bit happy with that, but again, we'll get a chance to consolidate if we need to later on. In terms of the ultrastructure, we're going to do a bit more detail on that, and then this bit we'll spend a bit more time on now in terms of exactly how muscle contraction occurs. Okay, so looking in a bit more detail. So again, here we go. If you have a look at this diagram, this is these are myofibrils, each of these from this diagram here. Look, there's your myofibrils, and each of these is a myofibril. What you can see, this blue area running across it, this is a new word, look. The blue area is called the sarcoplasmic reticulum. Okay, so that is effectively like what you've got in your normal cells, your eukaryotic cells. You've got your endoplasmic reticulum, but because in the muscle they're calling it the sarcoplasmic reticulum. And you can see how it's kind of extensive throughout all of the myofibrils. You've also got these things called T-tubules. The T stands for transverse in this case, and these run down here like that. So there's your T-tubules. Your sarcolemma we talked about before, look, and that's all around the outside, binding all of these together. That's your membranes. If you remember, this is one cell, one multinucleate cell. Alright, and then the myofibrils there we're talking about, your actin, your myosin and so on. Again, pay note of the mitochondria. Going to be lots of them. We're going to look at them in much more detail later on in the unit. So that's what your muscle fibre looks like. The filaments we looked at earlier on, if you remember, and what we're talking about doing in terms of creating this muscle contraction is shortening the sarcomere, shortening the distance between these two Z discs. How we do that in a bit more detail now, you can see there's the myosin heads, there's one myosin head. That's going to attach to this, and it's going to push it in that direction. Okay, ultimately the whole thing's going to shorten, like I said, like a concertina, and you're going to get this contraction. So at rest, So when your muscles are completely resting, the myosin heads are not attached at all. And the reason being they're blocked by tropomyosin. If you remember, the actin was the blue one, the thin filament, and you had another protein across it called tropomyosin. Calcium acts on troponin, which is attached to tropomyosin. Calcium acts on troponin, and that causes the binding sites on the actin to be exposed. And once that occurs, the myosin heads can then attach to it and then start moving it. So basically, the thing to take from that is that calcium is the key thing to starting muscle contraction. Without calcium, muscle contraction doesn't work. Calcium is ultimately released from the sarcoplasmic reticulum, but again we'll do that in a bit more detail shortly. Key thing, because it's a basic exam question they'll often ask, calcium is what is needed to initiate muscle contraction, because it binds to troponin, removes that, so the binding sites are exposed, which means that myosin can then bind to the actin. So how does it work? Again, if you have a look at the Prezi, there's some nice videos on there that'll show you it in a bit better detail probably than this, but this is the basics of what we're looking at. So calcium is released. Calcium will bind to troponin, which is bound to tropomyosin, and that removes them from the actin. In this diagram, confusingly, the actin is in red, so this is actin. Okay, and that'll remove troponin and tropomyosin from the actin binding sites, which allows then myosin to bind tightly to the actin. So there's a tight bond between the two there. Notice this 45-degree angle between the myosin head and the actin. You've got an ATP binding site here, and after calcium's removed, troponin and tropomyosin, the myosin head's attached. ATP is the next thing we're looking at, so if you just follow the arrow down here, ATP binds to its binding site in the myosin head, and what that causes is for myosin to be released from actin. Okay, so the ATP causes myosin to release from its rigor state, from its attachment to the actin. From there, here, ATP is hydrolyzed, and that forms ADP, an inorganic phosphate, which you already know about. That's your basic respiration, isn't it? So, well, basics from respiration and how ATP is used. That releases energy. So ATP hydrolyzed to ADP, and then what that causes here is for the myosin head to swing back. So from 45 degrees, it swings back to 90 degrees and attaches now, hence this is numbered, attaches weakly to the next binding site on the actin molecule. Okay, so it's gone from at 45 degrees, it's swung back to 90 degrees and bound to the next one. So if that's actin 1, this is actin 2, it's now bound there. And you can tell what's going to happen, it's then going to swing back to 45 degrees and ultimately move actin in that direction. Here we go. Inorganic phosphate is released. Once the inorganic phosphate is released, as we just said, this swings from the 90 degree to the 45 degree, pushing the actin in that direction as the diagram shows. Okay, so ultimately the actin's been moved along, and that's shortening the overall length of the myofibril. Finally, ADP is released. Once ADP is released, you get this tight binding state again. Okay, so they call it the rigor state on this, but it's tightly bound and waiting for the cycle to start again. Obviously what would happen next, ATP will come in again, it would release, swing back, weakly bind, push it forward until ultimately the distance between the Z discs is shortened and the muscle shortens too. Okay, this might go some way to showing you what it is I'm talking about. So if you have a look here, we can't see from that, there we go, that's your Z disc. Notice, you see why it's got its name now, because it's the shape of a Z. Your myosin here, your actin here. You can see the myosin heads. Now the animation is going to show you how overall the distance between the Z disc over here and the Z disc here shortens, because these myosin heads are grabbing the actin and pulling the actin in that direction, but likewise these are pulling them in that direction, and they're being pulled that way, and so on. So like I said a couple of times, you've got this whole concertina effect. Anyway, let me just get rid of all of these and let you watch the video.
Contraction of skeletal muscle tissue occurs when actin and myosin myofilaments slide past one another, causing the sarcomere to shorten. It is important to note that the individual myofilaments do not change length themselves, but instead overlap each other to shorten the sarcomere. Moving in for a closer look, we can see the details of how this occurs. The molecules you see here are myosin, which at rest has both adenine diphosphate and phosphate molecules attached at the individual myosin heads, actin, tropomyosin, and troponin. When we decide to move, calcium is released from the sarcoplasmic reticulum. These molecules bind with the troponin molecules. This causes the tropomyosin molecules to move, revealing myosin attachment sites on the actin myofilaments. The single phosphate attached to the myosin heads gets released as the heads move to connect with the attachment sites on the actin. The resulting formation is called a cross bridge. The remaining adenine diphosphate molecule is then expended in what is called a powder stroke when the myosin heads pull the actin inward. Once the adenine diphosphate attachment is spent, adenine triphosphate molecules attach themselves to the myosin heads. These molecules trigger the release of the myosin heads from the actin attachment sites and are immediately broken down into adenine diphosphate and a single phosphate as the heads move back into their resting position. This is called the recovery stroke. If calcium is still present, the cycle will repeat again, and the sarcomere will continue to shorten until the calcium ions are transported back into the sarcoplasmic reticulum and the muscle relaxes.
The diagram, quite a nice little picture actually. This is an electromyograph of a muscle cell, so you can see the kind of whole striped appearance on it. Anyway, I think that's about it really for today. So have a look at the question below, make sure you answer it, and then just be prepared with your notes and whatnot when you come into the lesson to crack on with whatever activity is appropriate for the knowledge you've gained.